Cool. All right, guys. I think we're gonna jump back in to get started. I just wanted to quickly introduce this next session. So, you guys are in for a treat because we've got three amazing scientists who are spending their professional careers focused on Castleman disease. We're going to kick off with Katherine Forsyth first. Katherine is one of our newer members of the Castle team, an amazing immunologist and scientist who we're so lucky to have thinking about Castleman's all the time. Then she's going to hand it off to Mike Gonzalez. Many of you all have met Mike before. Mike's an incredible bioinformatician who analyzes all of the complicated data that gets generated in the lab. And then I think Bridget's going to go third. Bridget Austin, who we introduced to you all earlier, and many of you all know, leads our biobank and our registry and our clinical trial. So she's going to be sharing some important recent findings. So with that, I will turn it over to Katherine for research updates. Thanks, everyone.
Hello. I'm shorter than David, so I'm going to lower that down. All right, hi everyone. I'm looking forward to telling you some of the research updates that we've been working on, and I look forward to meeting a lot more of you as the day progresses. So the goal of our team is really to connect improving clinical care with understanding the biology of Castleman disease, because we believe that when we better understand the underlying biology, that will give us insights into clinical care, and then how that clinical care works will then lead us back to the biology, and so on and so forth. So on our team, we both study the clinical data, so medical records. We have a whole team led by, you're going to hear from Bridget in a minute, but looking at maybe are there connections in the medical records that can give us insights into what's going on. And then on the translational side, we look at patient biospecimens and see if we can understand what's going on at a basic level. So we have many projects going on in all different areas of Castleman disease research, going from the cell types that are involved to treatments to clinical impacts. And in the last year, just to highlight two papers that we've published, we did a really deep dive to try and identify whether there was a virus or a bacteria or other kind of pathogen that was causing Castleman disease, specifically unicetric and IMCD, and we didn't see anything. So that's a really important finding because it's something that people have thought might be the case for a long time, and now we can really direct our focus in other directions. We also published a study looking at whether there were autoantibodies in patients with Castleman disease, and we did find some, which is very interesting to us because autoantibodies are often a feature of autoimmune disease, but we didn't find like one type of autoantibody that is present in a lot of Castleman disease patients, which would have pointed us to like a particular direction moving forward. So today, myself, Mike, and Bridget are going to tell some updates on some of our ongoing projects. So in the translational lab, I mentioned we use patient biospecimens. So what can we learn? So when we take the blood, which thankfully, thanks to some of you for donating today, we can identify what immune cells are present in the blood, and we can also identify what proteins are floating around. When we look at a lymph node biopsy, we can identify how many immune cells are present, what are the cells doing, so what are they making, which tells us a lot about what they think is talking to them, what is interacting with them, and then also where are the cells located. Cells, David mentioned, they like to talk to each other in the lymph node, and so knowing which cells are close to each other, which ones are talking to each other, can also be really important. And so together, all of this data points us to indicators of problems in IMCD as well as potential treatment targets. So the first story I'm going to tell you about is we were really searching for cell types that are, sorry, my contact is going crazy. That's a really unfortunate timing. Anyway, okay, everything's fine. Nothing to see here. That have both IL-6 signaling and mTOR signaling. So why do we think that's important? So as I'm sure many of you know, ciltuximab, which targets IL-6. And then there's this other drug, which David and others are on, sirolimus, which is effective in a subset of patients. And this targets a protein called mTOR. And then we also have a clinical trial that Bridget is going to talk about starting soon that targets an intermediary of these two called JAK. And so we were really interested in seeing if we could find cell types that are overactive for both IL-6 and mTOR, because this could potentially be a new cell type to target. And also these would be cells that are like overactive for both things that we know are detrimental and overexpressed in Castleman disease. So how do we do this? We used a lot of different approaches to try and interrogate this, and this is all lymph node data. So the first way we did this is we looked at a single cell level in the lymph nodes of IMCD patients to see what these cells are expressing. And so the cluster of cells that I've pointed to with the arrow, these are a cell called macrophages. And in the IMCD patients, you can see that they are darker red, so that means more positive for both mTOR and IL-6, and they're one of the few cell types that are up for both. We also were able to do a cool technique where we could stain a lymph node section with essentially a protein that is active when mTOR is active. And then we could take those regions that are mTOR active or mTOR negative and see what else is being expressed in those regions. And so we found that in the regions that are mTOR positive, they're also really high for IL-6. And this cell type called a macrophage was also coming out there. Then we did a really kind of cutting edge, very new technique that Joe really worked on quite a bit, where we can take regions along a lymph node at regular intervals, and we can pull out about 50 to 100 cells and see what are those cells making. And so we saw that in particular parts of the lymph node, macrophages were way higher. So macrophages is the bar, the IMCD bar is the pink bar that is much higher. So there are a lot more macrophages in IMCD lymph nodes compared to reactive controls. And then lastly, we could do some direct staining. So take, again, lymph node sections and stain them for proteins that identify macrophages, as well as IL-6 and JAK-STAT. And we saw that, indeed, the macrophages are expressing both of these. And so what does that mean for therapeutics? Well, we think that this provides additional evidence that our ruxolitinib trial targeting JAK might be efficacious because JAK is, again, an intermediary between these two pathways. It also makes us wonder whether a combination therapy of siltuximab and sirolimus could be effective in a subset of patients. And then also, now we have this cell type called a macrophage, which people haven't studied too much in relation to Castleman disease, but we think might be a really important player. So there aren't drugs that specifically target a macrophage, but there are drugs that target macrophage activation and certain subsets. So we think that that might be something we want to pursue in the future. The second research project, so David mentioned that your cells, your immune cells, are always traveling through the body, and then they stop in lymph nodes. And so we wanted to look at whether the cells are moving through the body correctly in IMCD. And so, so on the left-hand side, I have a little cartoon. So there's these proteins in your blood called chemokines, and these can tell the cells, hey, you should stop here, you should go here, really gives them like signpost directions. And so there's this one protein called CXCL13, and this tells the cells, go to the lymph node. And so the receptor for that is called CXCR5. And if the cell expresses that, it can essentially read the signpost and say, oh, hey, I need to go to the lymph node. So we looked in the serum of IMCD patients, and we saw way higher amounts of CXCL13. This is one of the highest hits we have in the whole study. And then in contrast, we see that the cells that express this receptor in the blood, there's fewer of them. So that tells us, hey, they're not in the blood, maybe they went to the lymph node, maybe they read the signpost. So we, again, performed this kind of regional transcriptomics in the lymph node so we could see where things were being expressed within the lymph node, and we saw that CXCL13 was, again, really high. And then we looked at a single cell level to see what cells are expressing the CXCL13. And so these are stromal cells. They kind of make up the kind of structural fabric of the lymph node. And you can see that, so the more red is the more expression, and you can see that in the IMCD, there's way more cells expressing CXCL13. And then lastly, we did some staining for cells, a particular type of immune cell that normally expresses really high amounts of CXCR5, so they're really responsive to this CXCL13 signpost. And so normally they're in this structure called a germinal center that some of you probably have heard about. And so we looked and we actually saw that there were fewer of these cells within the germinal center. So not only are the cells kind of traveling through the body weirdly, they're also not present where we think they should be present within the lymph node. And so this is work that we're writing up for publication now, but we think that this is an area that might lead to some potential new therapeutics in the future. So together we've used your patient biospecimens to identify a new cell type that we think is implicated in IMCD, and then also identifying that there's differences in how the cells are moving through the body that could also be playing a role in the disease. So with that, I'm going to turn it over to Mike.
All right. Can everyone hear me? Okay. Thanks so much, Katherine, for that nice presentation. And I'm happy to be here to be kind of the middle third of our sort of rapid fire research update portion of our summit. And so returning to kind of our overall list of projects, I just want to say that, my role here at the center, as David mentioned, is a bioinformatics scientist, which is what this really means is that I enjoy digging into these complex data sets, I enjoy crunching numbers, and I enjoy identifying signals in all of these separate projects that are pointing us in the same direction. And so the first project I want to share is how we were able to put together multiple streams of information, different pieces of the puzzle, to identify and target TNF signaling in IMCD. And they involve these 4 specific projects that you see listed below that are highlighted for you. And I'll individually get into each one and kind of share the piece of the puzzle that the results from that project helped lead us to that overall conclusion. And so the first signal we got about TNF, which is another pro-inflammatory cytokine, so you've heard of IL-6, TNF alpha is another pro-inflammatory cytokine that has a bunch of roles in our immune system. All of our immune systems produce it. But what I want us to understand about TNF specifically is that it's one of the first actors in the inflammatory response. So TNF starts getting produced, our inflammation goes up, and also TNF has very important roles in steering specific cells to where those immune cells should be going in the body. And so our first signal came from our serum proteome analysis. And you don't need to know what a proteome is, only to know that the blood samples that you guys generously donate, we're able to measure the amount of thousands of proteins from those blood samples. And when we compare it to control samples or patients in remission, for example, we can actually identify the proteins that are increased in Castleman. And when we do that, we can organize those increased proteins into specific gene and protein pathways that are driving certain biological functions. And so you can see our results over to the right, that IL-6 JAK-STAT, which is a pathway that we're very familiar with here with Castleman disease, but our second one is this TNF signaling, which kind of got our attention. And so next, we move to our single-cell RNA sequencing. So now instead of looking at overall proteins in the blood, we can actually pick individual cells from the blood and say, what genes are being expressed in that individual cell? And so now we have increased resolution to not only say, we still see this increased TNF signature in these single cells, but we can now say what potential cells are actually producing it. And so when we looked at that, you can see that T cells are really enriched for this TNF signature, and in addition, classical monocytes, which are the precursors of macrophages that Katherine was talking about previously. So now we have kind of 2 pieces of data that are pointing us to this exciting TNF signaling being involved in IMCD. And so now Katherine, Melanie, Abiola, a lot of our talented wet lab members start to ask questions in the laboratory. They say, how could this potentially be working? We can actually take samples that you guys donate to us. We can extract certain cell types. So if we're interested in T cells, for example, we can actually activate those T cells with the same amount of a given reagent. And we can actually see in our bar plot over to the far right that when we activate with the same amount of reagent with controls and IMCD cells, that a much larger proportion of those T cells become really active and start producing TNF. And so now we have 3 separate streams of information leading us to this TNF signature. And last but not least, the machine learning aspect. We're starting to leverage machine learning and AI models to actually predict drug disease pairs, specifically in Castleman disease and other diseases as well. And when we did this, we actually found that adalimumab, which is a TNF blocker, so it's a drug designed to block that pathway, was predicted to be really beneficial for Castleman disease patients. And so now we have kind of 4 separate streams that are all pointing us in the same direction, kind of where the rubber meets the road. Does this actually work in the real world? Can we give this to a patient? Does this benefit someone that's refractory to all other drugs that we might think of? And so Luke Chen is actually in the room with us today. Where is he? At the back. So we were lucky enough to collaborate with Luke Chen, who had a patient that was refractory to every drug that we could think of for Castleman disease, so siltuximab, sirolimus, and this patient just was not faring very well. And so Luke Chen approached our lab group and said, is there any Hail Mary that we can throw that might be helpful? And so we said, we're seeing this TNF signal in the data. It's never been used for Castleman disease, so it's a virtual unknown, but it is FDA approved for other autoimmune diseases. So you can give it a try. He prescribed it to the patient, and happy to report that this patient has been in stable remission for over 2.5 years and still counting. Very, very awesome story about how we put together the laboratory findings and then partner with physicians to actually try these alternative therapies and prove that they are beneficial. This was published in the New England Journal of Medicine, probably the largest medical journal on the planet. Please go and read it. It's nice and short. It's about 1.5 pages. It's free on the CDCN.org website. I encourage everyone to go and read that paper. And then the second project I wanted to focus on was trying to explain the underlying mechanism of what makes those immune cells hyperactive in Castleman disease. And I think now, and I do want to emphasize that this is more of a hot off the presses type finding, so this is unpublished, confidential, but it involves a number of projects that you see here. But I think a number of our results from these projects are pointing in the direction of dysregulated calcium signaling within certain immune cells. And I'll share why that is as we break down these projects. So first, our first signal came from what's called bulk RNA sequencing. So we can take those excised lymph nodes and we can actually measure genes being expressed in that lymph node tissue. And before I kind of get into this bullet point, is Kai Wen somewhere in the audience? Kai Wen, can you raise your hand? Kai Wen over towards the back, I did want to give a shout out to. In addition to being an IMCD TAFRO patient himself, he is also a very accomplished statistician who has dedicated his time and talents to working with us. And he is largely responsible for this analysis of the bulk RNA sequencing tissue. But what we found is we can identify those increased genes that are being expressed in that lymph node tissue, and we can actually ask a further question. We can say, okay, we have drug databases, and we can ask what drugs have the best chance of reversing the IMCD lymph node signature that we see. And so when we did that, I have highlighted there is isradipine, which is a calcium channel blocker being predicted to be effective. Now our second, very similarly with the single cell RNA sequencing data, we can now take that with the extra resolution of profiling those single cells. And again, we saw that a top FDA approved reverser of that IMCD single cell signature in the lymph node was verapamil, another calcium channel blocker. And my last piece of data, something that I'm really excited about, specifically as a quantitative geneticist. You may remember if you were here last year, I was able to share some preliminary pilot data about our somatic mutation search. If it's been a while since we've brushed up on our genetics in this crowd, just to remind you, so somatic mutations are a little different than what we classically think of as a genetic mutation. So usually we think of genetic variants that we are inheriting from our mom and our dad. These are what's called germline mutations. Now, somatic mutations are genetic changes that we acquire over our lifetimes. So think of something like cancer. So cancer is the classic somatically driven disease. And so usually, when cells divide in all of our bodies, there's some amount of somatic changes that occur. Usually these are benign. They don't happen within a gene or have any impact. But sometimes, if they occur in exactly the right spot and in exactly the right gene, they could have pretty significant downstream effects. And so I shared last year, I think, about data from 6 patients. I'm happy to report that we expanded this to 62 IMCD patients. And when we looked at the top somatically mutated genes, 4 of our top 5 were actually in calcium channels. And these calcium channels are really, really important in maintaining the intracellular calcium concentrations. And this interplay between calcium and maintaining that becomes really, really important in how these cells are activated as our immune systems deal with specific threats. And so this is a lead that we are definitely chasing down in the near future, but we are very excited about it. And then finally, just as a little teaser to leave you guys with, I think 2025 is really shaping up to be sort of the year of Castleman genetics. We were able to sign a contract with Regeneron, a very large DNA sequencing company. We've gotten the go-ahead to sequence up to 650 IMCD patients and their loved ones. So what we're really looking for here is ideally it'd be a patient and both parents, which would be classically considered a genetic trio. If you don't have that, that's okay. And you want to be involved in our genetic study, please find one of us. And we're happy to accommodate getting a blood sample and including you in this study. But we are currently awaiting genetic sequencing data. And yeah, so with that, I'm going to pass over to Bridget, but as she's walking up, I just want to end by saying, all of this work that I've just described this morning, and all of the work that Katherine described, and all of the work that Bridget's going to describe, it's only possible because we get the clinical information and the donated samples from patients and loved ones. Everything that we get from you guys goes directly into all of these experiments to answer questions about Castleman disease. And then second, I just wanted to mention, I am one of 3 people that is presenting this morning, but a lot of this work is reflective of a very talented team of scientists that are figuratively standing behind me. So wanted to thank you on their behalf. So, thanks.
Is the green button next? Yeah. Okay. Hi, everyone. Oh, is that better? Okay. Hi, everyone. I'm Bridget. I manage the clinical team at the Castle. If David didn't share, that's what we call our team here at the Castle. As many of you know, I think I've spoken to almost everyone in this room at one point, and as I just want to echo what Mike is saying, it's been an absolute pleasure to see you all year after year. If you guys remember, in 2021, we did a virtual summit, and that was my first summit, and I was like, oh, this is so disappointing that we can't, meet in person. So it's so great to see you all here. And a few of you have already donated blood this morning, so thank you to all of you and those who have in the past or will in the future. So I'm going to talk about some of our clinical projects. So as David mentioned this morning, we did have a recent publication come out about oligocentric CD. So a lot of you have probably experienced this in your own diagnostic journey, but diagnosis can be a very confusing time. It often takes a lot of experts looking at, a single lymph node to really determine if you may or may not have one of the many subtypes of Castleman disease. However, what we were finding is that there was a lot of anecdotal evidence of some sort of people that didn't really fall into the main 2 categories, right? We think about unicentric, the one region of lymph nodes, or multicentric. I think most of you here today know what group you kind of fall into, but there was this other group that was kind of in the center that we now call oligocentric. Clinically, it can present similarly to unicentric Castleman disease, but usually there are a few more areas of lymphadenopathies. So This was a really big finding, and it kind of echoes this whole sentiment of like Castleman disease is a spectrum. Even within your own subtypes, there's a lot of heterogeneity, right? You can't talk to two IMCD patients that are going through the same thing, or the two UCD patients that are going through the same thing. So I think this was a great finding by our team, specifically Sheila Pearson led this work. I think a lot of you have met her in the past, so I wanted to shout out her. Yeah, we love Sheila. Sheila used to be my boss. We love her. Yes, yes. But I also wanted to share some more recent work that actually was just submitted this week to one of the conferences we go to every year. We go to a conference called the American Society of Hematology. We call it ASH. So if you hear us say ASH, that's ASH. This year it's actually in Orlando. Fun. So the clinical team actually, throughout the team, I think we did, 6 or 7 total abstracts? I don't know if anyone knows that number off the top. Yeah, someone's nodding. Okay. Clinically, we submitted 3. So an abstract is like a short version of a publication, and it usually kind of previews what the whole publication could look like or could be like. So we submitted 3, and hopefully they get accepted. These 3 projects are really exciting. The first one is around pediatric IMCD. So we don't know a lot about what happens to children with IMCD. What are the best treatments for them, how they do on treatments, etc. So we sought to kind of elucidate that. Bless you. And this was actually the largest cohort we've ever had. So we reported on some pediatric IMCD patients a few years ago, but now we actually had 30 in this cohort, which is amazing. As you guys know, it's really hard to build these sample numbers when it's a rare disease. So we compared 30 children to 96 adults. And we found that they presented pretty similarly, except the children were actually more severe. And a lot of them had that TAFRO subtype that I think a lot of you have heard about. And they presented more severely, but they responded similarly to Castleman-directed therapies, so like siltoximab and all those other things. So although they present slightly more severely, they respond similarly. So that's one big finding from that. We also had another abstract submitted. So this was actually about IL-6 inhibition. So we've talked about IL-6 a little bit here, and a lot of you here today, I'm sure, have tried 1 or 2 IL-6 inhibitors. So we have siltoximab and tocilizumab. We are actually working with someone named Dr. Lu Zhang, who's actually visiting us starting tomorrow, I think. And he's one of our collaborators in China. And he discovered that some of his patients were responding differently to different IL-6 blockers. So, for instance, someone might start on tocilizumab and actually not respond, but then try siltoximab and have a response, which is interesting because those drugs can work very similarly. And in fact, in some cases, they're interchangeable. So why would you have a different response to one versus the other? So we looked in our Accelerate registry and also found a few patients in the same situation, and we reported on them. And we found that sometimes, for some people, we still don't know why, it's good to try both. If you don't do well on the one, you might do well on the other, which is a really big finding because some people just move on to something else before they try the other. So this is really exciting. We hope to do more work on this with Dr. Lu Zhang. He's at, I believe it's Peking Union Medical University in China. He's great. So yeah, that's that abstract. And lastly, this third abstract is really, by the way, that second abstract was led by Kayla McHugh. She's actually the one who's upstairs coordinating all the blood draws. She's wearing scrub pants. Some of you might have met her. She's great. Lastly, our third abstract is about kind of the diagnostic journey. So a lot of people, as you guys know, when you go through the diagnosis journey, it can be really unclear. And sometimes it can take months to even years to get a clear diagnosis. And we find that some of the folks in our registry may have been considered to have IMCD, but when our expert panel reviews these cases, they're not so sure. So we wanted to know, well, just because the panel thinks they're not sure about this case, like, how are they doing? Like, what's going on with them in real life? Do they still do well in these, Castleman-directed therapies? And we interrogated this, specifically Larissa. I know we've shouted out Larissa like 2 or 3 times, but Larissa did this project. And she found that they actually respond pretty similarly. So even if their diagnosis is a little unclear, you can still potentially benefit from one of these medications that we give to people who are like, yeah, that's a strong diagnosis. We really believe in that. So that's a really interesting finding, just because we know diagnosis can be so unclear. I also did want to briefly shout out some other projects that are in the works. Some of these are internal and external collaborations with a few folks in this room. There's a study on chemotherapy for IMCD. We're going to soon publish on sirolimus. So a lot of you guys know we did that sirolimus clinical trial last year, so we're soon going to publish on that. And then a few other studies comparing, TAFRO and HLH. HLH can be very similar to IMCD. And then we also have a case report coming out about breast implant-associated UCD, which is a new finding that we saw in one patient. More to come. Sorry? Potentially, yeah. Yeah. More to come. Yeah. Yeah, when in doubt. Yeah.
I mean, there's some adjustments I'd like to make.
Okay, so I think a lot of you have heard my spiel about contributing, and we're very grateful for those of you who have. I've even had so many people come up to me today saying, well, I want to give saliva too. I want to do this too. So like any of those questions you have, please feel free to come to me. My email is like all over the website. But just in case you didn't know, there are many ways in which you can contribute. First of all, you could donate your medical records. Every Castleman's patient in this room is eligible to sign up for Accelerate. And I believe almost everyone has, maybe everyone. So congrats to you if you have. Thank you so much. Yeah. As Mike noted, every single medical record is very crucial to this work. And some of you have thousands of pages, and each page is entered piece by piece by our analysts, and they're extremely crucial. It's long and hard work, but we've found a lot of our findings, like those 3 abstracts I just mentioned, were purely from your medical records. So thank you so much. Secondly, donate your blood. A lot of you have, especially if you're an IMCD patient, where we would really love your blood. It's really easy to do. You can either get it done locally at a Quest Diagnostics or go to your local physician appointment. Maybe you're getting an infusion. Usually they draw your blood beforehand. You can do that too. I think a few of us here have done one of those 2 things. If you haven't yet, email me and I'll show you how to do it. And of course, if you come to Summit, you can also donate. I will say we weren't able to draw blood from everyone today. We do have limited spots available, but I appreciate everyone who said they were willing to. Lastly, this one's a little bit newer. So if you're in the Accelerate registry, you're eligible to do a survey about pregnancy and fertility. Now, if you guys check your email, you've probably seen a bunch of emails about these surveys, and we have had a bunch of responses. But this is actually one piece of confusion. This is actually for men and women. So anyone who has tried to, get into the process of starting a family, you have started a family, you're eligible to fill out one of these surveys. And this information is really important because we're trying to get more data on, 1, the pregnancy and family starting experience, but 2, what do people do when they're pregnant? Do they stay on their CD medication? How does that go if they stay on it? Do they go off it? How do they feel when they're off of it? These are all really important questions, and I think it's actually been brought up by many of you that were interested in knowing how this goes. So if you're able to participate, we would love for you to. You can ask me or Larissa in the back if you're interested, and we can get you signed up for that. Oh. Don't think I mentioned this. That's why I have a little bubble at the top. If you're a parent or a sibling of an IMCD patient and you haven't donated blood or saliva to me and you're willing to, please talk to me because I would love to do that for the genetic study that Mike mentioned.
I've been getting the pregnancy and fertility emails. I am not an IMCD patient. Do you want the rest of us to also fill it out or just IMCD at this time?
I could talk to you after, but mainly IMCD at this time, but let's talk. Thanks. Lastly, I think David might have mentioned this earlier. I've been in and out. We do have a new clinical trial opening. I'm really excited to share this information. So previously we did a clinical trial for sirolimus, which was a pill. We're doing a similar type of trial for a drug called ruxolitinib. This is also a pill. It's a year-long trial. This is only for folks who have IMCD and have had a non-response to ciltuximab or tocilizumab. So if that's maybe you, feel free to come talk to us. This trial will be open later this year. It's not quite open yet. But just to answer, a couple general things about it, you're basically signing up to potentially receive this drug for up to 1 year. It's a pill, you take it every day, no infusions, which is great. You're signing up to come to Penn or a different site. We are hoping to sign up a couple more sites, but for right now it's just Penn. You would be coming for the first initial visit to make sure you're a good fit for the trial and all that. And then you'd also be signing up to come for your last visit. In between, there are a number of research activities you'd be asked to do, such as getting your blood drawn locally, getting imaging done locally, labs, all that jazz, which you're probably used to doing anyway, but it is, a little bit more work to be involved in a trial. So, oh, and you may receive compensation for travel as well. We try to accommodate those who are coming, out of town, taking flights, staying at hotels, all that jazz. So if this is something you might be interested in, feel free to come talk to me. We also have a few folks here today from the clinical trials unit at Penn. Do you guys want to raise your hands really quick? Hey guys. We're teaming up with, we're teaming up with the clinical trials unit to put on this trial. As you guys probably guessed, we all wear a lot of hats at the castle, so it's really nice to be able to rely on some other folks who are experts in trials. So we're really excited for that. So thank you guys for being here. All right, that's all I have. Oh, thanks. So we do have time for, do we still have time for questions? Great. If you guys want to come back up, the three of us can answer any questions.
Just a real quick question on the trial. If you qualify for it, are you guaranteed to get the medication or maybe a placebo?
No, you're guaranteed.
Okay.
You're guaranteed. Yeah, we don't have a large sample, a large enough community to do a placebo trial. Yeah, correct. Good question. Also, questions for any of us, not just me.
Any questions online?
We have one in the back. Yes, did you fill out the consent forms? Yeah. Oh, awesome. I will come grab you in a second.
Where do we go to get blood draw?
Good question. So if you're doing it today, like this gentleman is, I'm going to grab you in a second. We're going to go upstairs. Those of you might know we do the blood draws in hotel rooms just for, comfort and privacy. But usually you can also go to Quest Diagnostics and your local physician as well. Yeah. Oh, sorry. Just so the folks online can hear. Thanks.
Yeah, you just mentioned that medication that you might be doing the trial with.
Ruxolitinib, yes.
That's the one you mentioned for the oligocentric. Have there been clinical trials with that medication for the oligocentric Castlemans?
No, so oligocentric patients tend to present more like unicentric patients in that usually, this is not always the case, when you remove the lymph node, no further treatment is needed. Now that's kind of a case-by-case basis. So no, this has not been trialed with oligocentric.
Okay, thank you.
Any questions online?
Gary.
What put you on the trail of breast implant related onset? Because, and I ask this because I don't have breast implants, but a couple months before I first presented with Castleman disease, I had hernia surgery, and they put an implant in, where a hernia implant goes. And I think it was mesh and another element in there. So I'm just curious about that.
Yeah, that's a good question. I wasn't super involved in this case study, but as I mentioned, it's one patient, and I believe they saw the UCD pop up within a couple years of the implant. David, do you know more?
It came up shortly. Yes, it came up shortly after the implant was placed, and it was also in the armpit nearest, or well, I guess both implant, but it was armpit region, which is draining from the breast, and so that's why we think it's most likely secondary to the implant. Yes.
I don't know anything about that currently. If it's something you're worried about, I would talk to your doctor, but we haven't really published on anything else like that. Mm. Yeah, I haven't heard of anything prior to this one breast implant case, so I wouldn't know, but it's definitely worth bringing up. Oh, Dr. Chen, do you have an answer? You might know more. Dr. Chen, everybody?
I was going to say something. Some of the older breast implants can leak and cause stimulation of the immune system. So rarely there is a condition called breast implant-associated lymphoma. And Castleman disease is some form of lymphoproliferative disorder. So it's perhaps not an entire surprise that this can occur as a result of breast implants. But breast implants are quite different from other surgical implants, like surgical hardware. I don't think you would really not expect. Yeah, you would not expect it to happen there. Also, a lot of the newer breast implants, which have been used in recent years, don't have this leakiness. So I don't think anybody immediately needs to start panic about breast implants.
I was just going to say the same thing as Dr. Van Rees, and now I feel smart.
Okay. Any other questions? Okay, if not, yeah, feel free to grab any of us later. I believe we are breaking for 15 minutes.
So we're
Oh.
actually